This is a really nice summary figure that compares mitosis and meiosis in a number of different ways. So let's just take a look at the figure. Under meiosis, we have meiosis 1, and meiosis 1 is called reductional division because the homologous chromosome pairs are separated. So the cell that begins meiosis 1 is diploid. The cell that ends meiosis 1 is haploid. Why? Because again, homologous pairs have separated. Now, if the purpose of meiosis is to produce haploid cells, then some students will say, well, wait a minute, why do we need meiosis 2? The problem is, even though the chromosome number is haploid, each of the chromosomes is duplicated. And so you need meiosis 2 to separate the sister chromatids. That's why meiosis 2 is called equational division, because you're not decreasing the number of chromosomes anymore. All you're doing is separating the sister chromatids. So in meiosis 1, we have separation of the homologous chromosomes, reductional division, followed by cytokinesis. Then we have meiosis 2, separation of the sister chromatids to yield 4 haploid daughter cells that, if there was genetic diversity between the homologous chromosomes in the original cell itself, then there would be genetic diversity between these products of meiosis. And these are the haploid cells are called gametes. In the male, the gametes are sperm, and in the female, the gametes are oocytes or eggs. Now, let's take a look at mitosis. Remember, mitosis only has one set of division events. And in this set of division events, it's really equational division because there is no reduction in chromosome number. So you can see that mitosis looks a lot like meiosis 2, except for what? Except in meiosis 2, the number of chromosomes in each daughter cell is half. You see here, each of the daughter cells of mitosis is going to have 4 unduplicated chromosomes. Here, they're going to have 2 unduplicated chromosomes. So these are haploid daughter cells. These are diploid daughter cells. And these are genetically identical to the original cell that started the process. So at the bottom, we're looking at several processes: DNA synthesis, synapsis of homologous chromosomes, crossing over, homologous chromosomes lining up at the metaphase plate, sister chromatids lining up at the metaphase plate, and the number and genetic composition of the daughter cells. So in meiosis, both and mitosis, S phase of interphase is when the DNA replicates. In other words, both processes involve sister chromatids. Synapsis of homologous chromosomes only occurs during meiosis, and that's during prophase of meiosis 1, also called prophase 1. Crossing over, for our purposes of discussion, only occurs during meiosis, again during prophase of meiosis 1. Homologous chromosomes line up at the metaphase plate. Now, the homologous chromosomes join together or synapse and line up during metaphase of meiosis 1. The homologous chromosomes do not pair together and line up in mitosis. All of the chromosomes align along the middle. Sister chromatids line up at the metaphase plate. That happens during both meiosis and mitosis, but during meiosis it happens during metaphase of meiosis 2. During mitosis, it happens during metaphase. And then in terms of the outcomes, 4 haploid cells at the end of meiosis 2, 2 diploid cells at the end of meiosis. Now, just a point about this. In females, there are typically not 4 viable cells at the end of meiosis 2. Typically, there's only 1 viable daughter cell, and the other products are non-viable outcomes of meiosis. And we'll talk about what are called polar bodies in the chapter on human reproduction.